Hi, I'm Jack Daniels from the University of Southampton, and today I want to be talking about a recent piece of work which I've just completed, where we looked at how variation in personality traits can influence the barrier passage behaviour of invasive American signal crayfish. So this project was originally inspired by two papers which showed that personality could influence the spread of freshwater invasive species. Firstly, it was shown that mosquito fish, less social individuals are likely to disperse further, and in round gobies, more active individuals are likely to disperse further. And that's having a big influence on the spatial structure of the invasive population. However, both of those studies focused on continuous habitats, whereas modern rivers are heavily fragmented by anthropogenic barriers such as dams, weirs, and culverts, all of which can kind of prevent movements of a wide variety of species. So in this study, we wanted to look at how personality might influence the spread of invasive species, taking into account those fragmented habitats. We use signal crayfish as a model species because they're one of the most widespread and easily available invasive species in the UK. And also because there is an increasing management interest in using barriers to limit their spread. So there's kind of an applied aspect to the question as well. So to address this, we looked at two key objectives. So the first one was to characterize personality in signal crayfish by quantifying the repeatability of boldness, activity, and sociability, and looking at correlations between these traits to identify behavioral syndromes. And secondly, to look at how variation in these traits affects their behavior when presented at a model crump weir under laboratory conditions. So to assess their personality, we conducted 3 different personality assays in these rectangular tanks. The first assay we conducted was boldness. So crayfish were placed into the PVC pipe. You can see in the top image there. The front was covered. They were allowed to acclimate for about 5 minutes. And then a predation was simulated by rattling a pair of tongs in front. And boldness was measured as the time it took for them to emerge from the PVC pipe. Once they'd emerged from the shelter, their activity was measured as the number of lines they crossed as they moved throughout the tank in the subsequent 15-minute period. Sociability assays were conducted separately in a separate tank with a plastic pot at each end. A crayfish was placed in one pot and a rock of similar size and shape was placed in the other pot, and this is in the bottom image you can see there. The focal individual was released into the center in the neutral zone as it's labeled on the diagram. And then we recorded the amount of time they spent next to the crayfish in the pot versus the rock in the pot. And sociability was calculated as the log ratio between the time spent in the social zone and the time spent in the asocial zone. We did 2 repeats for each individual, and these were separated by a minimum of at least 24 hours. Once all the behavioural assays had been completed, they were allowed to recover for another 24 hours minimum, and then we placed them into a flume with a model crump weir as shown in the top right diagram. So we aimed to replicate the flow conditions at a real crump weir, which we successfully did. So the bottom diagram shows the flow. We had a crest velocity of about 0.75 meters a second, which represents a challenging barrier for crayfish, but one that they will be able to surpass. We then allowed them to move throughout this area for approximately 1 hour. And we measured 7 key metrics by monitoring their movements using a PIT system. So the 7 key measurements we took were the number of times they actually progressed onto the weir at all. Secondly, whether they actually attempted to pass the weir, with an attempt being classified as a progression into the area of high flow velocity, which you can see about approximately halfway down the weir face in the diagram. Secondly, thirdly, the number of attempts they made. We also measured the time it took for them to attempt after the beginning of the trial. We measured the maximum extent they made it up the weir face, also whether they successfully passed the weir, and the time it took them if they did successfully pass the weir. In terms of results, so the first objective we looked at was kind of characterizing personality, and we found that boldness and activity were both repeatable behavioral traits in signal crayfish. Sociability was not a repeatable behavioral trait. There was a lot of variation in that between trials. And we also found that boldness and activity were correlated, forming a boldness and activity behavioral syndrome. Conversely, sociability wasn't associated with either activity or boldness, but that's not necessarily a massive surprise considering it wasn't classified as a repeatable personality trait. Then in terms of barrier passage, we have a lot of graphs and key results here. So I'm just going to kind of focus on the main ones, which were that bolder individuals were more likely to attempt to pass the weir. And when they did attempt, they attempted more frequently, they attempted more quickly after the start of the trial, and they managed to move further up the weir face. Similarly, more active individuals were slightly more likely to attempt passage, and when they did attempt, they attempted more quickly. And again, sociability was not affected with any aspects of barrier passage behaviour, again, likely it being a result of not being a repeatable personality trait. The graph I presented here is just one of the key findings. It kind of illustrates the concept quite nicely. So on the y-axis is the number of times the crayfish attempted to pass the weir. On the x-axis is boldness, measures out the latency to emerge. So you can see that bolder individuals, which are those on the kind of left-hand side of the graph, which took a really short amount of time to emerge from the shelter, attempted a greater number of times. So there's several implications of this research. So firstly, as I mentioned, there's spatial structuring of invasive populations. So individuals with certain personality types are more likely to pass the barrier and lead the invasion front, for example. There's also implications for invasive species barrier design. It's really important to consider these aspects of personality when designing barriers to limit their spread. And there's also a substantial amount of crossover into fish passage research. There's a massive amount of variation in the effectiveness of different types of fish passage. And personality isn't really a factor that's been considered very heavily before. And this research shows that personality is associated with the ability of species to pass a barrier. So it's going to be an important consideration. So those are the kind of key results. I just want to reiterate the main findings. Firstly, that boldness and activity are personality traits. Secondly, that there's a boldness activity syndrome in signal crayfish. And finally, that boldness is a good predictor of signal crayfish behavior and anthropogenic barriers. I look forward to hearing your questions. And if you have any further questions after the time's up for this, feel free to contact me. I'd be really happy to hear from you. Thank you very much.